So in the previous video lecture, we imported our data into R and we structured it into a fileseek object. And in this session, we're going to use that fileseek object and get to the fun part and do our downstream analysis in R. And that's going to involve exploratory analyses. So we're going to make lots of plots. And then after that, we're going to do some differential abundance testing. So firstly, we'll get started on some theory on microbial diversity estimates. So bacterial diversity can be estimated for a single sample. So if we imagine we have three samples, A, B, and C here, you can see that for these three samples, they look very different. Sample A is quite diverse. It's got a lot of different genera. Sample B is somewhere in the middle, and sample C consists mainly of lactobacillus, so one taxa. So these three samples clearly differ in terms of their intersample diversity, and that's called alpha diversity. Now, if we want to instead relate samples to each other and see how similar they are, we look at something called beta diversity. So that's your between sample diversity. And on this plot here on the right hand side, we can see where each point represents one sample. You can see that certain samples are more similar and they form distinct clusters. So their underlying microbial composition data for points that are closer to each other are more similar. So alpha diversity matrix, it's a measure of within sample richness and evenness. So if we look at these two different communities, and each color represents a certain OTU or taxon, they actually have the same richness, meaning number of distinct taxa. So they each have 5 different colors, but they have different levels of evenness, which means in the community 2, we for instance have a much greater proportion of the blue taxon, whereas community 1 is much more evenly distributed between those 5 taxa. So why you think is alpha diversity useful measure for us to look at? And often in biological data, we'll find that these microbial diversity estimates are actually related to biological outcomes. And as an example, here is a figure from a study that was conducted at UCT, and this was on a mice study where they looked at pup stool alpha diversity, and the difference between the groups was whether the mum was treated with vancomycin, and if so, how long she was treated with vancomycin. And you can see that that clearly has a dramatic effect on the alpha diversity. So each one of these points represents one sample. So we'll see for each one of these samples, we get a specific numerical index of alpha diversity. So there's several alpha diversity metrics that we can choose from. Today I'm only going to discuss the most common ones that we'll be looking at in our downstream analyses. So first off, alpha diversity matrix can incorporate both richness and evenness, and that's really what we want from a good metric. So a commonly used one is Shannon alpha diversity, which places greater weight on richness, and then you also get Simpson alpha diversity, which on the other side places greater weight on evenness. And then we also get abundance-based measures of richness, which don't account for evenness, such as CHA1, and that's a non-parametric method used for estimating the number of species in a community. And they use singletons and doubletons to estimate the number of missing species in a community sample. And one thing that you should note here is that when you use data 2 automatically removes singletons as part of the ASV picking, because it's hard to distinguish between sequencing error versus actual biological variation. So singletons get removed, and that's why it's difficult to get an accurate CHA1 estimate, and we don't recommend using that when you have used the Data2 pipeline for your ASV table. Okay, so let's get some hands-on practice with our alpha diversity testing, and we can use that phyloseq object that we created in the previous session. And first off, let's have a look at the data and just see how many counts per sample we have and whether we can use all these samples that we have. So if we have a look at these reads, you'll see we get a number for each sample representing the total number of counts for each of the dogs. And what you'd hope to see is that they're all fairly equal with no outliers. The absolute amount will depend on your sample depth. It'll depend if you're using a MySeq or HiSeq platform. And so this cutoff of 5,000 that we're using here is fairly arbitrary. And in this case, we see that all our samples meet that criteria. But that would be up to you for your particular study to determine a logical threshold to use. So we mentioned that we need to normalize the sample read counts so that we can compare between the samples. So how we're going to do that is to first just convert our counts to relative abundance, and then to convert it back to count data, we're just going to multiply that those fractional counts with the median sample count that we determine here. So if we look at this total, that is simply the median read count calculated across all our samples. And then we're going to use this transform sample counts function, also from the phyloseq package. And there are two parameters that we need to specify. The first one being our phyloseq object, and the second one is a function. So this function we create here, and that will transform all our counts for all our samples. And in this case, we convert to relative abundance, and then we multiply by our median sample count and just round that number. So if we look at the number of taxa, we still have the same amount of taxa. And if we now look at our sample sums for the new phyloseq object called m.standard, you'll see that they have now all got the same total amount of read counts. So now we can move on and do our first alpha diversity plot. So again, there's a built-in function from the phyloseq package. You can actually pull up the help file for this to get some more info. So plot alpha diversity flexibly with ggplot2, and that's part of the phyloseq package. And you'll find a description, the usage, all the arguments that you can specify, and so on. But I'm just going to guide you through a simple example. So again, the first parameter is your phyloseq object, and then how you would like to place the different samples on the plot. You've got two parameters. The one is x, and the other one is color. So you can actually separate your variables on a horizontal axis, and you can also color with another variable. In this case, we're going to choose the treatment. So if we just look at that sample data for our phyloseq object, you'll see we've got dog, so we're going to place that on our x-axis, and then we've got treatment, so we're going to color by treatment. And then we can choose which alpha diversity metric we want to use. In this case, I've chosen Shannon, but you have a number of options here, including Simpson, Fisher, CHA1, and so on. So you guys can go playing around with that and see what difference it makes to your plot. And then the rest of the parameters that we specify here are just for plot formatting. So we specify a title, and again we use the paste 0 function, and in that title we can include our number of samples with the nsamples function, and we can choose the font size, whether it's bold or not, and so on. What you may have noticed is that this is a ggplot driven graphic. So you can add any ggplot type parameters here. So here, for instance, we have the plus theme, and then you can customize the plot however you want. So we have GM point. Those are all ggplot specific parameters. Okay, so let's run that. Okay, so now you'll see it runs, but that you get a warning message. It says estimate richness. The data you provided does not have any singletons. This is highly suspicious. So don't worry about that. That's what I mentioned in the presentation, that the data2 pipeline eliminates singletons, and that's why we won't use char1 estimate in this instance. Instead, we're going to use Shannon or Simpson. And for this issue, this warning, you can read more details in this GitHub issue here, but basically you can ignore that warning. So if we have saved this plot in the parameter called p, if you enter that, it should bring up the plot for us here on the right hand side, and here you can see we separated on our horizontal axis by dogs, B, G, and K, and then we've gone and colored by treatment. So you can switch this around, choose the variables that are most relevant to you in your study, and that's a good first look. So just from this, it seems that in general, dog G has higher alpha diversity than, say, dog K. Then we can use the PDF function to take that plot and send it to a PDF file. Then you can use send to your collaborators, publish, whatever. And when we do that, that'll print to your project directory in your R downstream analysis folder. So then we can look at this plot, and then it would be useful to know, are there significant differences between dog G and K in terms of alpha diversity? So for that, we can use the estimate richness function. We specify our phyloseq object. Again, we use Shannon diversity. So if we just run that and we look at this EST, it gives you the Shannon index calculated for each sample. So then, in order to do some statistical testing, we're going to combine this EST with our variable of interest, in this case dog, and we use the cbind, which stands for column bind, for that. And for that, to use cbind, you just have to make sure that you've got the same row identifiers in the two parameters that you're binding. So if we look at that, you can see now we have the Shannon diversity and the dog. And then after that, we can use the aggregate function, which basically summarizes our data for us, in this case, the mean for every dog. So let's do that. And you can see for dog B, G, and K, we've got a mean value, and it looks just from this that a dog G might have higher alpha diversity. But to test this, we're going to use Kruskal-Wallis test, which is a non-parametric rank sum test, so it doesn't matter if our data is not normally distributed. And then we're going to use temp, 1, which means column 1 and column 2, and we're going to compare by dog alpha diversity. So if we look at that data, we can see it's actually, there may be a difference by I, but that difference was not significant. And then just to show you how you can change the parameters for X and color, we can switch, so we use treatment instead for X and dog for color, and let's see how that turns out. So you can see now on our X-axis we've got treatment 0, 1, 2, 3, 4, and now we've colored by dog, so it just gives you a slightly different view of the data. So in the next video lecture we'll move on to the beta diversity.